for putting together this 2021 Biorex Aging Conference. I know we had the online version last year, and we were really all hoping to be there in person, especially my UK colleagues this November. Unfortunately, it's not been possible, but I think we have a very good program jointly between Israel and UK academics, which will foster many years of further excellence in aging research. With that, I'd like to introduce the first speaker of this session. And I'd like to remind all the speakers that we have 20 minutes allocated per person. I'm sorry, we're already running 5 minutes late. And to remind all of you that there will be an online and in-person mingling session at 2 p.m. this afternoon, 2 p.m. UK time, so that you can discuss these talks further. So that's 2 p.m. And you'll be given instructions how to join that session if we run out of time for questions. So do we have Dr. Illana Gozes from Tel Aviv University? I presume you will.
Thank you very much. I'm here. Thank you very much.
So thank you very much for being to speak. And your title is A New Look at Alzheimer's Disease, Diagnosis and Treatment Towards Healthy Aging. Thank you very much.
Thank you very much. So you heard already my first slide. So from a broad band of a new outlook on Alzheimer's disease, I'll actually go to a smaller detail and describe to you our work over the last 20 years, but I won't take 20 years to describe it. So 20 years ago we discovered a new protein to science. We looked to see how the brain protects itself, and we discovered a protein that we called activity-dependent neuroprotective protein, or in short, ADNP. And in short, without this protein, there is no brain. So it's a no-brainer. If we knock it out, there is no mouse. There's no brain. And it is a very important protein regulating about 400 genes during development and maybe more. And it does so by interacting, on the one hand, with the DNA. So it has an epigenetic effect. Everybody knows the word epigenetic. Not so many understand exactly what does it mean, but this is good for us. And the other thing, it also interacts with the cell skeleton. And you know, nerve cells have it's very important for nerve cells to have skeleton because they have a cell body and then very long processes that make the billions of connections that make our brain think. And so one take-home message that I would like to give you today is that we see similarities between abnormal development, namely in the autism spectrum disorder, and aging. And what is common that we discovered is actually the protein tau, which is tubulin-associated unit, but for us, it's Tel Aviv University protein, if you like. So tau deposits in Alzheimer's disease, and the more deposits you have, the more demented the person is. And tau also deposits, apparently, in neurodevelopmental diseases. So tau is an essential protein or an associated protein with the cell skeleton, the cytoskeleton, or the microtubules that I will describe today. And these are extremely important for the structure of the nerve cell and also for transportation of material in the nerve cell. So we have a nerve cell here, and then we have a long process that goes to the big toe in our foot. So ADNP is associating with the epigenetics, with the genes, the regulation of genes, and also with the skeleton of the cell. And what we saw, so one of our findings was that ADNP levels in the blood actually correlate with the IQ tests, so it correlates with intelligence, and it also regulates directly the major gene, risk gene for Alzheimer's disease that is called apolipoprotein E, which regulates cholesterol metabolism. Also, a colleague in Taiwan discovered that when he looked at complete serum proteomics, he discovered that ADNP was the only protein that went down in the serum of the Alzheimer's patients versus control. So it's a very important protein in the brain, but also in the body. And now comes another protein that I want to describe to you, and this actually is a finding of us from 2021. So we looked at another protein that is called SIR2-in-1, and SIR2-in-1 has been associated independently with healthy aging, and it protects against tau deposition. And we discovered a few years ago, actually, that SIRT1 is high, the levels of SIRT1 are high in centenarians, so people that live over 100, and decrease dramatically in Alzheimer's disease. And this is our very recent discovery. So we discovered that ADNP and SIRT1 actually interact in a complex, regulating the chromatin, so regulating epigenetics, and also they interact at the level of the microtubules to enlist tau to the microtubule. So you see on the slide, you see the microtubule shaft, and you see ADNP and SIRT1 interacting. And this we did biochemically, and we discovered that. And also, what we saw in Alzheimer's brain in comparison to control is a complete dysregulation of SIRT1, ADNP, and many other proteins that they interact with. So how does ADNP interact with microtubules? I have to share with you that I did my PhD on microtubules, so this is like my favorite. So I have here, like for all of us, is that microtubules are giving the structure, but unlike tubes, they actually are dynamic. So they give the structure to the cell, but in a dynamic way, and they are building on one side and kind of breaking apart on the other side and moving along the axon, along the nerve processes. And they are also essential for making synapses, for making the connection. So what we discovered that there is a small motif in ADNP that docks on proteins that are called microtubule end binding protein, and you see this here in silico. And actually, when it docks, it enhances other protein with the same motif to dock also. And this is like an orchestra, and the orchestra has to play, and there is a conductor somewhere also. The orchestra has to play and then move the microtubules forward, and this actually makes axonal transport and makes and is necessary for synapse formation. And this is just on a side. SIRT2-in-1 has a similar docking site on this microtubule end binding protein, which you see here. And SIRT1, this is in human neural cells derived from stem cells, from induced pluripotent stem cells, and ADNP and SIRT1 co-localize, and they are also co-localizing by biochemical technology. So mechanistically, and here I'm a biochemist by training, so I'm doing a little bit of biochemistry. So what I'm showing here is that this microtubule end binding protein, they bind to the end of the microtubules and make it move, but they also bind to the shaft of the microtubule, and then they enlist tau to the microtubule. And if you have ADNP there, you will get 20-fold more enlisting, which means it will protect against tauopathy, against tau falling off the microtubule and making neurofibrillary tangles, making these deposits that Dr. Alzheimer saw more than 100 years ago, which are associated, the more one has, the more cognitively declined the person is, and the more cell death there is. So this is the mechanism, and what we discovered is that this small motif in ADNP that we call NAP, that we took out from ADNP and made a small fragment, actually enhances this binding and may be developed as a drug. Another process that somebody got the Nobel Prize of, Japanese, and I can't pronounce the name, maybe somebody else in the audience, that microtubules are involved in, and ADNP is also involved in, is the process that is called autophagy, namely the cleaning of the cell from protein deposits. And ADNP doesn't only bind to the microtubule shaft, but in the end, but also to microtubule-associated protein 1, LC, light chain 3, which is a protein that undergoes lipophilization and makes the autophagosome membrane. So, and this is essential for the cell keeping healthy and cleaning from materials that are proteins that are depositing and are unnecessary. So ADNP is essential for this process as well. And NAP enhances ADNP. NAP is the small fragment from ADNP, the drug candidate, enhances ADNP binding and enhancing this autophagic system. And this system is deregulated in Alzheimer's disease, in Parkinson's disease, and also we discovered in schizophrenia. And what I'm showing you here is that ADNP and SIRT1 correlation is also abolished in Parkinson's disease. This is for Professor Nir Giladi, who is sitting here. And we can correct Parkinson's disease cell death with a fragment from NAP that we call SKIP, and this is in an animal model, and this paper was just published. And so ADNP is associated, as I showed you here, with Alzheimer's disease, with Parkinson's disease. I also said with schizophrenia because of the autophagy, and it also is associated with autism spectrum disorder. And so ADNP, like the last years, people have started sequencing children with autism within the autism spectrum, looking to see why are the children autistic, and ADNP is one of the major genes that is undergoing mutation, and this is the novel mutation. It occurs in the egg or in the sperm or during the initial steps of pregnancy, and then the parents are okay and the child is autistic. And here you see 35,000 sequencing, and here you see 46,000 sequencing, and still ADNP is a lead. And just, so the side track here from aging, I'm looking at the beginning of life, and this is, so the ADNP syndrome is an autism intellectual disability. It's a rare syndrome, but there are many that looks a little bit like it. And what we discovered, stemming from this discovery that there are mutations that occur out of the blue, I actually thought that it might be interesting to see if there are mutations that occur in our brains, I mean, not ours specifically, but in the Alzheimer's brain, and maybe these could drive part of the disease, because as you know, Alzheimer's disease is totally sporadic. And the major risk factor is actually aging, and we want healthy aging. And what we discovered is that ADNP indeed undergoes mutations, and you see many different mutations here, depending on the person. And it's somatic mutation, so it's not inherited. It occurs during life, and we know now that this mutation actually accumulates with aging, and this is in the brain. And we saw a correlation between the accumulation of mutation and the degree of tauopathy, the degree of tau deposition, which is associated, as I said, with dementia. And we also saw that in Alzheimer's disease, actually in amnestic mild cognitive impairment, NAP was tested actually clinically. This you see here in the lower part of the slide. You see it here, and it actually protected short-term memory. And ADNP is not the only protein that undergoes mutation in the Alzheimer's brain. We saw in the aging brain. We saw many other genes. As you see here, hundreds of genes are undergoing mutation in all brain areas. And this is something that I would like to follow up. But what is interesting is that we, of course, in science, you are never alone. And there were when we were doing our work, there were two other groups that were doing the same work. And then a third group took the three works together. And what was very interesting is that there were similarities between disparate groups around the world, finding mutations in the same kind of genes, very long genes. And also what we saw is that the genes that were mutated are associated either with the cytoskeleton, so now you all know what I mean by the cytoskeleton, and the microtubules are major part of the cellular skeleton, and also with autism, and also with intellectual disability, which is like, you know, the autistic world. And here, this is from another parallel work that was done, one of the works, and they showed that the pathways culminate in a pathway on tau, and also in a pathway that is regulated, that is called ACT. The protein is called ACT, which is associated with growth. And we have shown that ACT is a major pathway that is affected in the ADNP syndrome. So ADNP regulates ACT. So again, we tie these very important proteins that regulate, and they regulate each other. The mutations are not only found in the brain, but they are also found in the blood. So this is another recent work from 2021 where we showed mutations in the blood of people that are suffering from post-traumatic stress syndrome, which is a major risk factor for Alzheimer's disease. And again, here we saw many cytoskeletal protein genes mutations, and also in the immune system. So tumor necrosis factor alpha, which is a major immunoregulator, was specifically mutated in PTSD, and ADNP is associated with stress and this is a study in Israeli soldiers, and we saw a correlation between ADNP and corticosterone. So it's not only Alzheimer's disease, it's also that has somatic mutation, but there was a paper very recently in biological psychiatry that I was asked to comment on, and also in schizophrenia there are also mutations, and these mutations are associated with the glutamatergic transmission, and the glutamatergic transmission is affected. This is the excitatory transmission. So this is necessary for learning and memory. It's affected also in schizophrenia. It's also affected in Alzheimer's disease. It's also regulated by ADNP, as you see here down. You see that with ADNP, with intact ADNP, there is a staining of the glutamatergic system complete. And then if there is half the content of ADNP in the mouse, and this is in the hippocampus, the area of learning and memory, you see a marked reduction. And when we treat the mice with NAP, this short fragment of ADNP, we correct it. So also in schizophrenia, there are somatic mutations that may drive the disease, and it might also occur in Parkinson's disease. Here I'm showing you our last work it's now in press in biological psychiatry, and what we found here is, again, in a model of autism for ADNP mutation from the autistic children, the major mutation of the autistic ADNP kids, which is kind of cutting the protein into half, it's heterozygous mutation, and a major pathology that we've seen is the Alzheimer's pathology, the tau pathology, but this appeared in very young mice, like it appears in the children, which I'll show you in a minute. And what we also saw, we did complete RNA sequencing of the spleens of these mice, and a major gene that is regulated only in the females, by the way, is FOXO3. And FOXO3, like sirtuin-1 and even more, has been a gene associated with healthy aging because it protects the microbiome, it protects the autophagy process, and it also is associated binding with the sirtuin-1. And again, also in these mice, ACT1, as I said, is regulated by ADNP. And the tauopathy in this mouse, the tau deposits, was actually associated with electrical activity. That was done in collaboration with Dr. Moran Rubinstein, an electrophysiologist, a young scientist, an excellent young scientist in our faculty. And actually the work was done by an MD PhD student, Gidi Carmon, who was just finishing. And what we discovered is that there was change in the visual evoked potential of these mice. And this is the abnormal situation. And when we treat for 4 days with NAP, we got immediately the precise normal situation. And this is the tauopathy that we see in a postmortem analysis of an autistic child. And here is a control. And when we put a NAP in tissue culture and we measure the binding of tau to the microtubules, we see a dramatic reduction if there are mutations. And by the way, this is the mutation that was very prevalent in Alzheimer's, somatic mutation. This is the most prevalent mutation in the ADNP children. We see here a dramatic reduction. So where am I going now? 2 directions. One, we would like to do a clinical trial in the ADNP children with NAP, and the other one, together with a group in Hong Kong, we got the Catalyst Award just recently to actually use an artificial intelligence way or paradigm to actually discover new drugs for Alzheimer's disease based on the somatic mutations that we have discovered. So I would like to thank my students all over the years, and you are going to meet Maram Ganaim. She is doing something else, but I don't have time to discuss today. Thank you very much.